Hello viewers. Today in this program, we are going to learn about the antibiotics. We will specify our learning to the antibiotics which are bacterial cell wall inhibitors. Now there are many antibiotics which can inhibit or prevent the bacterial cell wall synthesis, and out of all, we are going to study about the beta-lactam group of antibiotics. In 1800, synthesis or the research work in microbiology started when the people or the scientists came to know that microorganisms can cause disease. After this, a research work was going on, and in 1928, there was a British scientist who was working in St. Mary's Hospital in London, and his name is Alexander Fleming. Now he came with an astonishing result in one of the nutrient agar plates. What he observed in his nutrient agar plate is when he was trying to grow the Staphylococcus aureus, which is a bacteria, the growth was inhibited due to presence of a green mold. Now in this picture we can see the presence of the mold which inhibits the growth of the bacteria. So Alexander Fleming was the one or the first one to postulate that there is some chemical secreted by the fungus which inhibits the growth of the microorganism or the bacterial cell. After that, the two terms were known in the science. One is antibiotic and the other one is antimicrobial compounds and antimicrobial agents. Now let us see what is the difference between the two. When I talk about antibiotics, that means I am referring to a chemical which is produced by a microorganism that kills or inhibits the growth of another microorganism. That means an antibiotic is of microbial origin. On the other hand, the antimicrobial agent is a chemical that kills or inhibits the growth of the microorganism. Now this antimicrobial agent need not be an origin from the microbial cells. It can be any chemical compound also which inhibits the growth. That means in other way we can say that antibiotics are antimicrobial agents, but all antimicrobial agents are not antibiotics. Antibiotic function because they have got antimicrobial activity. Now this antimicrobial activity is of different types. For example, the antibiotic may inhibit the cell wall formation. This cell wall formation I am referring to is the bacterial cell wall formation. The second property or the second mode of action of the antibiotic may be it may inhibit the protein synthesis. The antibiotics may also inhibit the replication of the DNA, or there are many antibiotics which inhibit the synthesis of the RNA. The synthesis of the essential metabolite may also be inhibited by the antibiotic. Now these essential metabolites are nothing but the primary metabolites. That means the central metabolites which are needed by the microbial cell for their growth, for their reproduction. However, the antibiotics are the secondary metabolite. So in other way we can say that a secondary metabolite inhibits the growth of the primary metabolite, and therefore the whole metabolic pathway is being degraded. The beta-lactam family of antibiotics prominently have four prominent groups. These groups are penicillins, cephalosporins, monobactams and carbapenems. Now why I said penicillins? That means this group has got numerous penicillin molecule. It is a group, it is not a single antibiotic I am referring to. And under each group we have got numerous antibiotics. Now how they work, what is their chemical structure and how they inhibit the growth of the different microbial cell, that we are going to learn today. The beta-lactam group of antibiotics that we talk about have got a prominent structure, and that is why they are called beta-lactam. This is due to the presence of the beta-lactam ring. Here in this slide you can see that there is a presence of this beta-lactam ring, and along with this beta-lactam ring some other ring may be attached. Now this is not necessary for all the beta-lactam antibiotics. I will refer to the antibiotics when I go to the detail. Okay, but the beta-lactam ring should always be there, otherwise it is not a beta-lactam antibiotic, and the presence of this ring gives the antimicrobial property to this group of antibiotics. The second thing is, to the beta-lactam ring at the carbon 6 position, here you can see we have a formation of a side chain. Now, substitution of this R group which is present in the side chain brings about different antibiotics which have got different pharmacological properties, and therefore they can be used for different infectious diseases which a human suffer from. Going on to the specific group, that is penicillins. Now let us first see the structure of the penicillin. Now penicillin is a beta-lactam antibiotic. That means it should have a beta-lactam ring. Here you can see in this slide it has got a beta-lactam ring, and along with the beta-lactam ring it is attached to another ring which is called thiazolidine ring. Now this thiazolidine ring attached to the beta-lactam ring is specific for the penicillin molecule, and therefore this is called the general structure of the penicillin. The substitution of this R group which is at the side chain brings about different molecules of penicillins. For example, we can have penicillin G, we can have penicillin V, we can have methicillins or different other types of penicillin. Basically, penicillins are of three types. One is natural penicillin, the biosynthetic penicillin, and semi-synthetic penicillin. The natural penicillins are the one which is naturally produced by the microorganism. That means we are not dictating the microorganism to produce a compound. The microorganism produces it naturally without addition of any precursor or without addition of any side chain precursor, and that is a natural penicillin. The example is, here you can see the natural penicillin example is penicillin G. In the case of biosynthetic penicillin, the fermentation, that means the antibiotic production which is carried out in the industry, takes place or it is controlled by adding the side chain precursor, so that we have production of only one type of penicillin. Now in the natural penicillin, numerous types of penicillin may be produced, and out of this only benzyl penicillin is effective. In order to inhibit the production of other compounds and only to produce only one type of penicillin, the industry goes in for the production of biosynthetic penicillin, that is addition of side chain precursor is done in the fermentation, and we have production of only one type of penicillin. In semi-synthetic penicillin, what is done is the natural penicillin or sometimes the biosynthetic penicillin are taken. For example, penicillin G or penicillin V is taken. It is chemically or enzymatically split to form 6-APA. That is, the basic structure of the penicillin remains, because it is due to this 6-APA it has got the antimicrobial activity. So 6-APA when attached to some other group makes a derivative of the penicillin, and this is called the semi-synthetic penicillin. Here we can see the semi-synthetic penicillin like ampicillin, oxacillin, then methicillin. They are all semi-synthetic penicillin. This pink box you can see over here is the main structure which should be there in the penicillin molecule, otherwise it will lose its antimicrobial activity. So this is the pink box you can see in this structure, and all the structures have got a pink box, that is the basic structure of the penicillin, and then we have the substitution of the R group, and therefore numerous penicillin molecules can be produced. We said that beta-lactam group of antibiotics are the cell wall inhibitors. Now let us first see that what do the microorganisms have in their cell wall and how this cell wall formation can be inhibited. Basically, we say that prokaryotic cells are of two types. Either they are gram-positive or they are gram-negative. In the case of gram-positive, here you can see in this slide, this structure or this portion of the gram-positive cell which is towards the outer side is quite thick and rigid. This is peptidoglycan. This peptidoglycan is found in the gram-negative cells also, but this is very thin. And this is deeply embedded because after peptidoglycan there is another layer which is lipopolysaccharide, or which is a lipid present in the gram-negative bacteria. So what happens? The gram-positive bacteria have a peptidoglycan which is the supermost or the uppermost layer, while in the case of gram-negative this is deeply embedded. This peptidoglycan is the major component of the bacterial cell wall, and penicillins or the beta-lactam antibiotic target this peptidoglycan because peptidoglycan is not found in the eukaryotic cells. That means the human do not have the peptidoglycan, or the host cells do not have the peptidoglycan, and therefore the antibiotics specifically target the peptidoglycan layer only, which is present in the microbes or in the microorganism. So let us see the structure of peptidoglycan and how it is synthesized. If I go in the detail to the structure of peptidoglycan, I take an example of a gram-positive bacteria, that is Staphylococcus aureus, because peptidoglycan is much more thicker and exposed towards the outer side in the case of gram-positive bacteria. Now here you can see the peptidoglycan has got a basic backbone structure where there is presence of N-acetyl glucosamine, the red disc you can see over here, and the green disc are the N-acetyl muramic acid, and they are present in alternate manner and are joined by beta-1,4 glycosidic bonds. Now this backbone of the peptidoglycan shows the presence of the amino acids. There are 4 amino acids, or rather I should say that there is presence of 5 amino acids which is present, and this is attached to N-acetyl muramic acid. And this amino acid chain is again attached to another amino acid chain which is coming on from another N-acetyl muramic acid, and these two peptides are joined together by the presence of a linkage which is called pentaglycine bond. Here you can see the structure of the pentaglycine cross-linking which is joining the two peptides together, and these two peptides attached to the N-acetyl muramic acid. So let us first study the synthesis of peptidoglycan, since peptidoglycan is a target site for all the beta-lactam antibiotics. During the synthesis of the peptidoglycan, we find that N-acetyl glucosamine condenses with UTP, uridine triphosphate, to form UDP N-acetyl glucosamine. This is the formation of UDP N-acetyl glucosamine. UDP N-acetyl glucosamine reacts with PEP, that is phosphoenol pyruvate, and there is formation of UDP N-acetyl muramic acid. The 5 amino acids are now added to N-acetyl muramic acid. These 5 amino acids are alanine, glutamate, lysine, and 2 molecules of D-alanine. So these 5 amino acids which form a short peptide is attached to N-acetyl muramic acid. Going on further, the muramic acid or N-acetyl muramic acid pentapeptide. Why we call it pentapeptide? Because it has now presence of 5 amino acids with itself. So it is muramic acid with pentapeptide. Now this is transferred from UDP to dolicol. Here you can see in this picture that it is transferred from UDP to dolicol. Another N-acetyl glucosamine is added to this molecule. Here you can see the structure of the pentapeptide with muramic acid and then addition of the N-acetyl glucosamine. Now why this N-acetyl glucosamine is added? Because after muramic acid you should have N-acetyl glucosamine, then muramic acid, then N-acetyl glucosamine. Like this the backbone has to be formed. Okay. So after the addition of the N-acetyl glucosamine, the 5 glycine molecules are now added to the lysine residue. I told you there are 5 peptides in this slide. You can see that the first one is your alanine, the second one glutamate, and the third one is lysine. Now to this third one there is addition of 5 glycine molecule. So again a pentapeptide is formed, and this pentapeptide, which is only formed with glycine residue, forms the pentapeptide bridge between the 2 amino acid or between the 2 peptides which is coming out from the muramic acid. Okay, so after this what happens is the whole disaccharide decapeptide. Why do we call it decapeptide? Because now we have 5 peptides over here, or the 5 amino acids over here and the 5 amino acids over here. So 5 and 5 makes a 10 amino acid peptide, and therefore we call it a decapeptide. Now this decapeptide is now added to the non-reducing end of the existing peptidoglycan molecule. Here you can see that it has to join, or it is added to another decapeptide which is formed to the opposite end. So the existing peptidoglycan, which is called the primer, has to be added to it. So how this takes place? This takes place with the help of a reaction which is called transpeptidation. That means an enzyme transpeptidase is essential for it. It is the one which forms the pentaglycine bridge between the two peptide molecules which is coming off from the muramic acid. This takes place with the help of transpeptidase. What happens in transpeptidation is you have the removal of this terminal alanine molecule. I told you in the terminal portion we have two alanine molecules. So the terminal alanine molecule is removed and to this alanine molecule this is attached. So pentaglycine linkage is being formed between the two between the two peptides which is coming up from the muramic acid. This reaction is called transpeptidation, carried out by transpeptidase. The action of the beta-lactam antibiotic comes over here, the last step of the peptidoglycan synthesis. This transpeptidation reaction is being inhibited by the beta-lactam group of antibiotic, whether it is penicillin or any other beta-lactam antibiotic. It inhibits this transpeptidation. So what happens is the bridge which gives the rigidity to the cell wall is not formed. So they remain fragile. The cell wall synthesis does not take place completely. The cell wall does not mature. The peptidoglycan is not formed. So ultimately what we see is you have a penicillin which attacks the bacterial cell, and ultimately the bacterial cell synthesis does not take place and the cytoplasmic content which is present in the bacterial cell comes out. Also sometimes the spiroplast formation may take place. How this penicillin works, or what is the exact mode of action of this penicillin, let us see. The penicillin has got a strong affinity for transpeptidase. That means the penicillin attacks the transpeptidase. The function of transpeptidase is inhibited and now we have formation of an inactive transpeptidase. Here you can see the penicillin which forms an inactive transpeptidase. The function of a normal transpeptidase is, as I told you, is to form the pentaglycine cross-linking structure or the cross-linkage which has to be formed. Now this transpeptidase, when forms the cross-linkage, you have the formation of this and the two amino acids which is coming off from the backbone of N-acetyl muramic acid, they are joined. But if this transpeptidase has or is attacked by penicillin molecule, these are the penicillin molecule which has got a strong affinity for this transpeptidase, which is now called the penicillin binding proteins or PBP. And when it binds to it, it becomes inactive, and when it becomes inactive, this reaction does not take place and therefore the bridge is not formed between the peptide and transpeptidation is inhibited. The peptidoglycan synthesis does not take place. So this is the actual mechanism of the synthesis of the peptidoglycan and how this biosynthesis process can be inhibited by beta-lactam that we have learned. But the microbes are very clever. They are also developing as the human beings are developing, and they are finding out new therapy to overcome them. How they are clever? Because they are developing many mechanisms which could inhibit the effect of penicillin, and these are called the resistance mechanism against the penicillin. What are these? Number one is destruction of the beta-lactam ring. I told you the beta-lactam ring is very essential to have the antimicrobial activity. And if this destruction of the beta-lactam ring takes place, the penicillin becomes ineffective, and that occurs by the production of an enzyme which is called beta-lactamase, which degrades the beta-lactam ring. So the microbes produce beta-lactamase which degrades the beta-lactam ring. The second mechanism by which they can develop the resistance is altered affinity for penicillin for their target site, which is the penicillin binding proteins. I told you the transpeptidase is the penicillin binding protein to which the penicillins bind. So if the penicillin binding protein could be altered, so the penicillin does not bind and therefore the penicillin becomes ineffective. The third is the decreased penetration of the antibiotic to the target site. This is specially seen in the case of the gram-negative bacteria, because in the case of gram-negative bacteria, the peptidoglycan is embedded deep inside after the lipopolysaccharide and it is very thin. So, decreased penetration also results in ineffective penicillin activity. Second group of the beta-lactam family of antibiotic, and that is called cephalosporin. Now this is also a beta-lactam antibiotic. That means it also have the beta-lactam ring. Here we can compare the two structures, cephalosporin with penicillin. The presence of the beta-lactam ring, which is also there in the penicillin, is same, but the difference is the presence of another ring. Now this presence of ring, which was thiazolidine ring in the case of penicillin, is replaced by another ring which is dihydrothiamine ring in the case of cephalosporin. There are different generations of cephalosporins. For example, we have the first generation of cephalosporin that came out early, and this is absorbed from the intestine, which is useful against the gram-positive cocci and certain gram-negative rods only. However, there is second generation of the cephalosporins which act against, they have got a much wider range of antimicrobial activity, because now this second generation of the cephalosporin act against the gram-positive cocci as well as numerous gram-negative rod. This was acting against certain gram-negative rods in the first generation, while in the second generation we find that numerous gram-negative rods are inhibited by the action of the second generation of the cephalosporins. The third generation of cephalosporin, which takes into account the very common name which is used today also, is cefotaxime. Now they are the one which act against the gram-negative rods, and they are used for treating the diseases of the central nervous system. And the fourth generation of the cephalosporin, which is cefepime, have improved activity against the gram-negative bacteria, which are mainly involved with the urinary tract infection. Now, why this development of the generations of the antibiotic took place? Because we have the resistant strains which are coming up. The first generation cephalosporins became ineffective. We have the resistant strains, and therefore the numerous strains have to be come up. Now, how these were developed is, let us see this diagram in the slide very clearly. In the presence of cephalosporin, we have got a beta-lactam ring and dihydrothiazine ring. Now there is a side chain, as I told you, this is called R1, and there is another side chain which is R2, which is over here. The substitution of R1 and R2 bring about different molecules of cephalosporin. For example, the substitution of R1 can bring about all these cephalosporins, and substitution of the R2 group brings about all these different types of cephalosporin. So, simply by substitution of the side chains, we can have different generations of the antibiotic, because we have the resistance variety that is developing every day. Another group, which is the third group of beta-lactam family of antibiotic, is monobactam. Now, monobactam, if I go to the structure here, you can see clearly in this slide, monobactam also have got a beta-lactam ring. But this beta-lactam ring is alone. It is not attached to any other ring. There are many side chains, but there is no other ring, and therefore we call it beta-lactam ring is alone and not fused to another ring, in contrast to the other beta-lactam that we have studied, that is penicillin and cephalosporin. They work against the gram-negative bacteria. There are few names are given. For example, the nocardicin A is very common monobactam antibiotic. Now, it interferes with the bacterial cell wall synthesis. They are all beta-lactam. They all inhibit the bacterial cell wall synthesis. But in the case of beta-lactam, which is monobactam, we find that the binding to penicillin, they inhibit the cell wall synthesis by binding to penicillin binding protein 3 or PBP3 of the aerobic gram-negative bacillus such as Enterobacteriaceae and Pseudomonas. The carbapenems are the last group which belongs to beta-lactam family of antibiotics. In the case of carbapenem, we see that they have got a broad spectrum of antimicrobial activity and they have a structure that render them highly resistant to beta-lactamase. Now, beta-lactamase is an enzyme produced by the microorganism in order to overcome the effect of the beta-lactam antibiotic. So now we have a generation of an antibiotic which is carbapenem. It again has got a beta-lactam ring, and to this beta-lactam ring you can find that's presence of a side chain R1, then we have another side chain R2 and another side chain is R3. So substitution of R1, R2 and R3 by different compounds bring about different antibiotics which belong to the group carbapenem. This carbapenem interferes with the bacterial cell wall synthesis by binding to penicillin binding protein, which are essential for the cell wall synthesis. So basically what we see over here is beta-lactam group of antibiotic are the group which prevents the bacterial cell wall synthesis by targeting a specific component in the bacterial cell wall, and that is peptidoglycan. The last stage of the peptidoglycan synthesis, that is transpeptidation, is being inhibited because transpeptidase becomes the penicillin binding protein as the molecule binds to transpeptidase, makes it ineffective, and so peptidoglycan is not synthesized. So this is the effect of the beta-lactam family of antibiotics. Thank you.